And then what's significant about tomorrow evening at 11:59 p.m.? Right, so before you take out another subject to study tomorrow night on a Friday evening, make sure you study this class first and do the quiz, right? And the good news is if you're preoccupied with something else at 11:59 p.m. on Friday evening, it will automatically submit as long as you save your answers. Okay? This is where we left off. One more announcement. What's significant about the next time we get together on Tuesday? We have our first exam. Okay?
Will you be posting a study guide?
Yeah, so we're going to have a study guide and we'll have a practice exam. And those will probably start going up tonight. Okay? By tomorrow you'll have them. And SI sessions as well. Take a look at the posting of the SI sessions. We are working on getting a larger classroom for SI sessions, especially before this exam, because a lot of the classrooms won't fit the number of students that will show up. So we put in that request, and we're optimistic that we'll hear in time to be able to have a bigger classroom for a review session for exam 1 on Tuesday. So pay attention to the BD Learn announcements because they're going to push out relevant information over the next couple of days. Okay? Anything else? Okay. Why did the dog cross the road twice? Right? Why did the dog cross the road twice? Because he was trying to fetch a boomerang, of course. That is my Alyssa's handwriting, by the way. So, that's a 9-year-old who wrote that one. What does a peanut say when it sneezes?
Cashew.
Cashew! Alright. I'm pretty sure it's not like that too. So organelle is going to be fair game next Tuesday. And as homework, I want you, and I will put the answer on BB Learn, but I want you to put these in order in the cell. You can use your book. The cell is a protein factory. Put the following organelles and cell parts in the order in which they're involved in protein synthesis. So let's give you a hint. Where are you going to start? With the nucleus. That'll be the first thing. Okay? Some of you are like, yes. Okay? Awesome. There's only like 12 more items to put in order. So go over that, and I will send out in an announcement the answer. All right, that brings us to cellular respiration. And we're going to finish out cell bio. We're going to get into histology and tissues. We will start that, but we won't ultimately finish it. And so the remainder of histology and tissues, the remainder of Chapter 5 will be on the next exam, not for Tuesday. Okay? So it doesn't mean we're skipping anything. We're just going to go as far as we get today. And that's where I will stop writing questions. Okay? Any questions about that? The exam will be, nobody asked the question about, what's the format of the exam? Right? Are you guys curious or you don't care? Okay, fine. We'll just move on. Oh, no, you want to know? Oh, okay. So it'll be 50 multiple guess, or multiple choice, depending on how hard you study. I do put in my test questions A and B only, all of the above, none of the above, or B and C only. You may see in the occasional question that that's the format. There will be, somewhat like 5 to 10 questions that will show up on the screen. And there will be images, and those images should be recognizable from the lectures. Okay, it's not going to be some obscure image of a dandelion in my backyard that you've never seen before. Okay? And I ask some, wonky question about it. It'll be stuff that you've seen, as long as you've studied with your eyes open. Okay? Those will also be multiple choice. We have an hour and 15 minutes. There'll be 2 different versions of the exam. We'll have a number of proctors. All right, that's basically to keep the honest people honest in all reality. should have no trouble finishing it within an hour and 15 minutes. Okay? In fact, most people will be done well within an hour. But that means that you're going to have to ensure that you use that time wisely. Maybe go back and look at your Scantron and make sure you didn't do any transposition errors, because if you make a transposition error, that's on you, and I will not give you credit for it. Because once I hand back the Scantron, I don't know when that transposition error did or did not take place. You with me? So I'm just telling you ahead of time, you'll have plenty of time. But if you come up to me afterwards and say, oh, I circled it correctly on my test packet, but I bubbled in the wrong one on the Scantron, bummer. That's a real bummer. I will have extra Scantrons, okay? No extra charge. Comes included in your course fees, in your registration, your tuition dollars, extra Scantrons. Isn't that nice? So use them. If you are worried about the erasure mark, get another Scantron. You'll have time to redo it. Okay, but I cannot and I will not give you credit back for making bubbling errors. Okay? By now in your career, you should be very familiar with how to bubble correctly. Okay, so if you are a problem bubbler, maybe practice over the weekend. Okay, YouTube it. How to make a good bubble on a Scantron. Okay? All right. Cellular respiration is where we're picking up here. And what I want you to be able to do as another piece of homework is I want you to be very familiar with glycolysis, where it happens, Krebs cycle, and the electron transport. So if you have this slide, why don't you either circle or write or put a single asterisk in this weird-looking finger, which is also referred to as a mitochondria. Okay? My daughters think that looks like a finger. This is a really gross finger to have. What's wrong with it? It's pink and energy, babe. So that is a mitochondria. Put the asterisk next to where glycolysis happens, a double asterisk next to the Krebs cycle, and 3 asterisks next to where the electron transport chain activities take place. So do that now. See if you can get this accurate. Okay, you ready? Should we unveil the answers? All right, so glycolysis. Where does glycolysis happen? Where does the asterisk go for glycolysis? In the cellular cytoplasm, right? So it's inside the cell, but it happens just within the cytoplasm soup, the intracellular fluid. The Krebs cycle now, where does that go? That goes within the mitochondria. So you're seeing this glycolytic pathway here is outside of the mitochondria, but it's inside the cell because the mitochondria is contained within the cell. Krebs cycle happens within the mitochondria, and the electron transport chain, where is this happening right here? That's in the, what? Intermembrane space, right? Between those two membranes that the mitochondria has. Okay, so, back to sort of this conversation that we had at the beginning of the semester, the thought process is this inner membrane was the plasma membrane of that cell as it previously existed before it was engulfed by the cell to have energy-making capabilities in the cell. And that's where the second membrane came from. Okay? So, as homework, what I do want you to understand and appreciate not only is where this location takes place of these different energy sources, but also to understand that you can generate ATP from proteins, carbohydrates, as well as fats. You create unique intermediates from these compounds, like amino acids that will feed into the citric acid cycle, glucose, which will first go into glycolysis and generate acetyl-CoA, which will go into the citric acid cycle, otherwise known as the Krebs cycle, otherwise known as the tricarboxylic acid cycle. Remember, 3 names for the same cycle. Or you can actually have fats. And fats in the format of glycerol will be manufactured into pyruvate, and then acetyl-CoA, and then ultimately to the Krebs cycle. So from any one of these starting organic materials that we've looked at, we can generate ATP. Now, what's the difference? Lots of homework for you. And I'll send out the answer for these as well. I want you to be able to define glycolysis, glycogenolysis, gluconeogenesis, and glycogen. You can go ahead and do it right now. Write them down. Write down the answers to the definitions. Who's still writing? Okay, a couple. So we'll start at the top, because those who are still writing probably down towards the bottom. Oh. Glycolysis. What is glycolysis? Who has a definition of glycolysis? What are you guys writing down, man? Your fantasy football picks from last week? What's that?
Breakdown.
The breakdown of glucose. Exactly. Breakdown of glucose yielding ATP. That's the only other thing I'd add to that. How about glycogenolysis? Okay. Is he the only one that knows the answers? Okay. Right here. You're correct, by the way, but I hope they didn't hear you yet.
The breakdown of glycogen in the liver creating glucose.
Okay, so the breakdown of glycogen to form glucose. They can then go into glycolysis and then into the Krebs cycle. Gluconeogenesis. How about somebody in the back? Back of the room. Sorry, you're doing a really good job, but I'm trying to give everybody a ribbon here. Anybody in the back? Gluconeogenesis. Break down the word. Anybody on that side? Here.
Yeah. It's converting lactic acid back to glucose that can be used again with or without the presence of oxygen.
Okay, so it's actually generating newly formed glucose. Okay? And you might utilize an intermediate, like a byproduct such as what? Lactic acid. Okay? So you're generating new glucose from scratch. Glucose, new, beginning. That's how it literally translates, right? Gluconeogenesis. Glucose, new, beginning. And then glycogen. What's the definition of glycogen? You want this one?
Isn't it a bunch of glucose binding together in the liver for storage?
Yeah, it's a bunch of glucose monomers strung together into a polysaccharide, which is glycogen, and that's basically the storage form of glucose. Okay? It's found in the liver, it's also found in muscle tissue as well. Okay, I'm just making a note for myself to post these answers. All right. So we just kind of did this on our own, comparing our definitions. Now here's an example type of question that you might see on Tuesday. You perform a chemical analysis of a bone cell called an osteocyte. I have to tell you what that is because we haven't covered it yet. And you find that it contains large amounts of pyruvate. So you determine that what has occurred? You determine that which of the following has occurred. How many of you say gluconeogenesis? Okay, how many of you say the Krebs cycle? How many of you say glycogenolysis? How many of you say two of these? Okay, a couple of hands. How many of you say none of these? Okay, so the none of these has it, and they are correct. Why? What's that? It would be glycolysis, right? Because that's what pyruvate is produced by. It's a byproduct of glycolysis, so none of those are correct. Okay, so that's kind of an example of how you might see sort of this A and B only or none of these types of questions. Make sense? Is that helpful? Give you a little hint as to how I write questions? Cell biology, passive membrane transport. So if we look at membrane transport, we can either do it with no energy or with energy. If you do it without energy, it's considered passive. And it goes down what we call concentration gradients. So you have something that has a high concentration, it's going to move from an area of high concentration to low concentration. This is review, all still in review. We will get new material today in histology and tissues. So inside the cell, the extracellular concentration, that's what those brackets mean. This is nomenclature for concentration. So extracellular concentration does not equal the intracellular concentration. Okay? This is how biologists like to express mathematics, right, with words, and then a symbol. Okay? So whenever the extracellular fluid equals the intracellular fluid, what is probably happened? Equilibrium. And what's the state of the cell most likely? What's that? Is it alive or dead, I guess is my question. It's probably dead. Because the cell is going to work really hard to pump certain things out and pump certain things in. So the inside and the outside are never equivalent unless the cell is actually probably not working anymore. So the question is, what direction will solids move? They're going to move down their concentration gradient, which is a way of saying they move from an area of high concentration to an area of low concentration. And we refer to this movement as diffusion. Diffusion. So this requires no energy. It's passive transport of materials. So if we have passive transport like diffusion, there's no energy needed. It goes from high to low. You just need to have a channel open for ions to move. Well, a second way to do this is you actually utilize energy, typically in the format of ATP, and you move things against their concentration gradient. And so you go from low to high. You actually build up a stockpile, and you add more wood to your stockpile of wood as you get ready for this coming winter. Energy is required, because when you're piling up that wood, you're sweating. You're using energy in order for that to happen, so that makes sense. If it goes from high to low, no energy is required. But go from low to high, to stockpile more of that ion requires energy, typically in the format of ATP. Now, if we look at sugars, ions, and amino acids, they move either by passive or active transport, depending upon what the concentration gradient is in that particular tissue. In other cases, you've got things that move via what we call bulk transport because they're too large to just simply flow through a hole in the membrane of the cell. You actually have to bring them in, and we'll talk about that. Usually this happens using vesicles. We call that endocytosis, okay, or penocytosis in the case of very small molecules. So here's an example picture. The extracellular fluid is up high, and the intracellular fluid is down low. The extracellular fluid has a high concentration of these red-looking ions. We'll call those sodium. And so when this channel right here, which is a transmembrane channel, when it opens, which direction do these sodium ions want to move? They want to move from high to low, so they move into the cell. And that's what you're seeing right here, is it went from 2 little lonely sodium ions to 3. If you want to go the other direction, would it be passive or active? Active. It would be active. Very good. Now, we have some unique cases of diffusion, specifically called osmosis. So simple diffusion works just fine for small lipid-soluble substances because that means they're lipid-soluble, they can move through the membrane. But in some cases, the solutes are too large to actually move. And so what instead moves is water. And when water is moving, we call this osmosis. Now, lots of students have had this before, but it's inevitable that students struggle with this concept on the first exam. So I want you to pay close attention. Osmosis is a special type of diffusion that refers to the movement of the water, not the solute. But it is leveraging the fact that you have a diffusion gradient in place. So no energy is required for osmosis. So here is a picture of a little experiment. And let's pretend that you have side A, or what we call, let's say it's cell A, and right next to it is cell B, or side B, and this dotted line represents a semi-permeable membrane. So these are two adjacent cells that are touching each other. And you're going to see how solutes or water would move from one cell to the other. Well, the water molecules are represented by these little blue spheres. And the large orange spheres are albumin. We talked about albumin. What was albumin? It's a protein. Where do you find it? Find it in the blood. It's one of the main proteins within the blood as a soluble protein. And the purpose of albumin is shown right here. Well, if you have high amounts of albumin inside A, which direction does albumin want to move? It wants to go from A to B, right? But it's too big to fit through this membrane. So it cannot move. So instead what happens is the water molecules move to dilute out the albumin as best it can. So the water molecules go to an area of high concentration of albumin to try to lower, essentially, the concentration of albumin. Make sense? Because there is no way that this large albumin sphere is going to fit through the semi-permeable membrane. So instead what happens is water comes across, and you increase the volume of water separating out the albumin, and the albumin never leaves that particular reservoir. This is an example of osmosis. And this is exactly what happens in your blood vessels. And this is why you have fluid in your tissues that wants to come back into the venous circulation, because there's lots of albumin in the venous circulation. Make sense? So the fluid in your tissues is reabsorbed via osmosis back into your bloodstream, and then it's available to go back to the heart, and it's available for the next cardiac cycle. Okay? So this is actually considered what we call osmotic pressure, right? Where you've got the osmotic pressure of water increasing as more water comes over to dilute out where the albumin is. So the water, the terminology is the water moves and follows the solutes because the solutes are too large to actually passively diffuse. So if we look at osmosis, we can see another example within the blood circulation that's shown here. The water moves down its concentration gradient until it reaches equilibrium. Well, water concentration depends on the number of solutes that are found within it. So we can have either three different situations. Something could be hypertonic solution, it could be an isotonic solution, or it could be hypotonic. If it's a hypertonic solution, that means the salinity, the saline content, the salt content is higher than normal. If it's isotonic, that means it's physiologically appropriate. That's usually a 0.9% sodium chloride solution would be an isotonic solution. Next time you're in the hospital, if you see an IV bag, look at the label. It will typically say 0.9% sodium chloride solution. That's why you deliver a saline bag rather than pure water. So let's explain this. If it's hypotonic, which would be pure water, right? Then you would have no salt within that solution. So, three different erythrocyte conditions in your red blood cells. Isotonic solution is the middle one. That's a red blood cell put in an isotonic solution. It's supposed to have this little dimple. And it has a dimple, it's biconcave, it has a little dimple on both sides. If you look at it on the edge, it looks like an hourglass. And that's what it's supposed to do. Now, what do you think, what solution do you think the one on the far left was put in? Hypotonic, that's pure water. And this sucker is swollen, right? Because it's in pure water. And so where's the water going to want to go? Into the cell. And it'll go so quickly that it could rupture and burst the erythrocyte. So if you gave a patient pure water in an IV drip, you actually could kill them. Okay, so it's kind of important to pay attention to this segment. Okay? Especially if you're going to be my doctor someday. I will test you before you touch me. Okay? What about this one? What kind of solution was that red blood cell put into? It's the only option available, guys. You have almost 100% chance of success here if you were listening. Hypertonic solution, right? So which way is the water going to want to move? If there's lots of salt, this is a solution, hypertonic, it's like a 2% sodium chloride solution right here. Which way does the water want to move? Out of the cell. And so it crenates the cell. It shrivels down. That's called crenation. Alright, so what would this look like next Tuesday maybe? Maybe something like this. This red blood cell, this erythrocyte, can be put into which type of solution? Isotonic, hypertonic, hypotonic, or I cannot determine from the information given. Don't laugh. That could be real sometimes. Okay? Which one is it?
B.
B, hypertonic solution. Very nicely done. You guys are A+ students for Tuesday, I can tell. What is facilitated passive diffusion? So in this case, a facilitated passive diffusion uses a carrier molecule, a transporter. It utilizes these pores or these channels to bring things across. This was the example that I was showing you earlier of sodium coming across utilizing a channel. So in the case of our erythrocyte question, we weren't talking about channels, so we just have water moving quickly. Now, when we look physiologically, we can set up these different concentration gradients and we can purposefully open a gate when we want to. And so doing this is how we establish an electrical difference from one side of the cell to the other side. Primarily the outside of the cell to inside the cell. Because if you have sodium, which is what charge? +1. Sodium is a positively charged ion. It's a +1. And if you have lots of sodium outside the cell, and you have less sodium inside the cell, just in that example of sodium, is the cell going to be negative or positive with respect to the outside? It's going to be more negative. If you open a channel and sodium rushes in, now what happens to the electrical potential inside the cell? It becomes more positive because sodium rushes in carrying a +1 charge. Right? That is electricity, guys. That's how actually you send an electrical impulse down a nerve. And we're going to talk more about it. It's more complicated than just that. But those are essentially the nuts and bolts of it. So if that makes sense to you, you're in good shape. Okay? If you're confused now what I'm talking about, and you still don't understand what these yellow things are with this giant purple structure, then you need to do some studying before Tuesday. Okay? So these channels can be gated. They're usually closed. You can trigger them to open by actually physically touching them, mechanical triggers. You can trigger them to open using a ligand, something binds to it. Or you can trigger them to open using electricity. Okay? So facilitated passive diffusion, these guys are facilitated by this carrier molecule. All right, here's another type of question that I think is helpful to look at for next week. You've got a cell that has a 10% glucose solution inside of it. Its intracellular environment is 10% glucose, 10% sugar. If you put it into a beaker environment that was 20% glucose, what's going to happen? Is it going to swell, shrink, or no net change? What's that? It's going to shrink. Okay? C. If you put the 10% glucose cell into distilled water, question number 2, what's going to happen? It's going to swell. This is the same question as the red blood cell question. It's just asking it a different way. And then, third condition, 10% glucose into a 10% glucose, what's going to happen to the cell? No net change. Make sense? Okay. Sweet. Let's look at moving things that are large. Okay? And doing it using energy. So active transport. Active transport, this is getting more complicated now, right? Now you're like, uh-oh, there's more than just like, sodium going on here. Well, the movement of solutes against their concentration gradient requires active transportation. This is the sodium-potassium ATPase that you guys know and love. I know you have a drawing on your mirror in your bathroom of sodium-potassium ATPase, right? If you don't, you should. It comes out really nicely, actually. Okay? And Windex takes it right off. But that's a great place. Windows, mirrors, great place to study. You just draw stuff with a whiteboard marker. Okay, Sharpie almost there, right? It'll come off and throw me off if you use a Sharpie. This requires energy because you're needing to input energy into the system in order to do the work. It's like rolling a ball uphill. Takes energy. Just to let it roll downhill, you just drop it and it goes. These proteins are sometimes called pumps. So that's why we call this also this sodium-potassium ATPase. We also call it the ATPase pump. Or the sodium-potassium transporter. Right? So what's going on is, if you follow the orange line, we're going to pump out 3 sodiums every time this thing runs, and we're going to pump into the cell 2 potassiums. So we're transporting things in and out at the same time, using energy to do that. And this pump, it runs in all cells of the body. Have anybody ever felt a pump while it's been on for hours? What does it feel like? It gets hot. It's warm. This is a pump in our body that's running non-stop, and it actually generates a significant amount of heat. This is what kind of helps you keep your basal metabolic temperature at about 98.6 degrees because of these pumps. Okay? So it is a mechanical pump that's running. Active transporter, we could also have one like called secondary active transport. Hang on, I'm just going to figure out where I am. Secondary active transport. This uses an indirect ATP consumption. So for example, if you look down here on the basal side of the cell, you can see the sodium-potassium pump is running. Right? You see little sodiums that's shooting out, right? And you see that it's generating energy or using energy, ATP becoming ADP and PI. Every time you cleave one of those phosphate groups, you actually liberate energy. So it's burning energy, it's running energy. These pumps, it's like it has to be plugged in. Right? You don't have a pump that you know of that doesn't require either electricity or mechanical movement. Right? If you have a bike pump, you have to sit there and, pump it this way or pump it this way. Or, if you have a compressor, you plug it into the outlet. It takes energy in order to run it. So this is running, creating a high concentration of sodium outside the cell. Now look on the apical surface. Glucose, this is a sodium glucose transporter. Okay, the SGLT. This SGLT is going to bring in sodium down its concentration gradient, and glucose just hitches a ride. And this brings glucose into the cell after a meal, utilizing the gradient that you've set up using this pump at the basal surface. So this is still considered active, but it's called a secondary active transporter because it's dependent upon the gradient that's established by this active pump. Make sense? So you're basically creating a gradient, and then cleverly you're utilizing that gradient, and you're hitchhiking the ride, glucose comes in. So it's a symporter. It's still active because that whole nomenclature. ATP is still consumed even though it's indirect. If we move on to study a little bit more about the sodium-potassium ATPase pump, and this is going to be very important later in the semester. You can see down here, it says this is what keeps you warm in the wintertime. This uses about 30 to 50% of all your body's ATP that you make just to run the pump. Very inefficient, to be quite honest. But what it does is it maintains a resting potential. A potential means a change in electrical difference. So it has a higher, what? Inside. Higher potassium concentration, abbreviated with a K. That's from the periodic table. That's why people, why isn't it abbreviated with a P? I don't know. It's always been a K. Okay? Higher sodium concentration outside, Na. Na is the abbreviation for sodium. Also from the periodic table. Okay? This is going to be very important as we continue in the semester, because if you look here, if I take 3 positively charged ions, the sodium is Na+1, I send 3 positive charges out and I only bring in 2 positive charges in, what's my net difference? 1. It's not a trick question, right? It's 3 minus 2. 1. I know we're biologists, but you can use your phone if you need to. So it's a difference of 1, and it's a loss of a positive charge out of the cell, so it creates a slightly negative environment inside the cell. And this establishes this resting membrane potential inside the cell being slightly negative. It usually sits at about minus 70 millivolts. Millivolts, okay? So 1,000 millivolts will equal 1 volt. Right? So volts are what comes out of the wall. Okay? And our body uses the exact same type of electricity. It's just back way down. Because you're talking about moving small little molecules that have these positive charges. Okay? All right, bulk transport. So these are kind of cool pictures. These are electron micrographs. And these are the artist's drawing on the bottom. And you can see this is a clathrin-coated pit. A clathrin-coated pit. What the heck is that? Okay, these are pits within the membrane of the cell. And clathrin itself has receptors that recognize cholesterol. So this is how a cell will bring in cholesterol, is using this clathrin-coated pit. So you can see that the receptors are localized to an area. They find the cholesterol molecule, and then they start to invaginate. The pit drops down, and then it's going to pinch off on the inside and bring in this vesicle that contains cholesterol inside of it. It's like a little balloon inside the cell, this vesicle that contains cholesterol now. And then it can go to its various locations in the cell and be utilized. Okay? This process right here is called endocytosis. So this is the way a particle that's too big to cross the plasma membrane, that's what PN stands for, then it uses proteins. And these proteins are these receptors. Endocytosis. Okay? If it's packaged in a vesicle and it's a large vesicle, we actually call this phagocytosis. And that refers to phago meaning eating. So this is cellular eating. Phagocytosis. Penocytosis is cellular drinking, so it's when the molecules are smaller. Now you can reverse this process, and you can actually take a vesicle and have it fuse with the membrane and dump the content, and that would actually be called exocytosis. That's how you get rid of certain things. Or you manufacture something inside the cell, and you say, you know what, this product needs to be outside of the cell, so I'm going to put it in the vesicle, it's going to fuse with the membrane, and it's going to dump out the cell. Okay? You're looking at gut epithelium. right, in lab. Intestinal epithelium, right? Stomach epithelium. And there's a structure called the goblet cell. What does the goblet cell have inside of it? Mucus. And if you look at the histology and you capture it at a point in time, you can see where the goblet cell has fused and it's exocytosing and dumping out mucus. And in the lining of the stomach, the reason you dump out the mucus is you coat the inside lining of the stomach with this mucus layer so that the hydrochloric acid that you manufacture to break down your food doesn't actually break down your stomach. The mucus is like this coating that protects you. Okay, kind of cool. So that would be an example of exocytosis. All right, any questions on this particular lecture before we move on? Yes?
So, that example of endocytosis with the pits, is that only applicable to cholesterol? Does it have one?
No, so the question is, the cholesterol-coated pits, is it only to cholesterol? That's an example that's cholesterol specific. But, for example, phagocytosis happens by macrophages all the time, like right now. So macrophages are cruising around in your body, finding debris, bacteria, and engulfing it in the same type of way. It doesn't use collagen. It uses other types of proteins, but it's the same type of process. Okay?